how do monoclonal antibodies they act as drug carriers? How are they named? Let's get into all this mechanics today. Welcome all to Is Pharmacology Difficult podcast. I'm your host, Dr. Radhika Vijay, MBBS MD Pharmacology, and this is the audio hub to get the best, simplified basic tips, strategies, methods, and lots of ideas to learn better, understand better, and make your concepts crystal clear. If you really find and if there's a question hovering in your minds, is pharmacology difficult? Lend me your ears for a while and let in the magic of knowledge. Products of immunization, they are the polyclonal antibodies. Now, why poly? Prefix is added. Let me explain this. There are multiple epitopes or natural antigens, and these epitopes, they are known as antigenic determinants. Now, there is a generation of separate clones of activated B lymphocytes. On the other hand, if a single clone produces antibodies, then they are directed towards the single epitope. And when such things happen, then such antibodies will be known as the monoclonal antibodies, very famously abbreviated as capital M, capital A, small b, and small s, MAPS. For large-scale production, Hybridoma technique, it is adopted. And what is that? When specific B lymphocytes, they are fused with tumor cells. Which tumor cells? Mouse myeloma cell. What we obtain that are known as the somatic cell hybrids and especially referred to as the hybridomas. This keeps and maintains the abilities of both the fused parts, B lymphocytes. They have the antibody forming nature and capacity, while mouse myeloma, they can proliferate endlessly. Hence, production of monoclonal antibodies is continued and they can be purified for the clinical use. I hope you got the whole mechanics and the whole great concept. Did you? Only drawback of the murine or the rodent monoclonal antibodies or MAPS is their short half-life and their ability to produce anti-mouse antibody allergic reaction. Yes, they can do it. So they should be used with a lot of caution. So one way to cure this problem is to partly humanize these antibodies simply by genetic manipulation. Can we do that? Yes, we can. Then they are proudly stated as chimeric antibodies. Chimeric. What do you mean by saying chimeric? That is, they contain some genetic manipulated material which is not a pure, neither of mouse nor of human, but chimeric, mixed one. Got it? Their special feature is the coupling of murine variable region to the human constant region in the finally obtained structure of antibody. Now you got the meaning of the term chimeric. Combination of the variable region of the murine part and the constant region of the human part. Now, one good quality of totally humanized monoclonal antibodies is their very low antigenicity or antigenicity. Okay, it depends on you, whatever you call it, but I hope you got the meaning of this whole thing. Low antigenicity is obtained by recombinant DNA technology or by other techniques. Yes, there are many more techniques like grafting of complementarity determining regions, which are famously abbreviated as capital C, capital D, capital RS. Now, these are the regions of the murine monoclonal antibodies on the human immunoglobulin framework. Are you getting the whole structural part of it? Actually, that makes a lot of sense at this particular point when we are discussing all of this genetic manipulation. You have to really imagine what is fixed on what is coupled with what, and how are we obtaining the chimeric things or the hybrid things. That is so important. Now, this whole antibody structure talk was actually a little complicated. Yes, I do agree with you. But no worries, you know why? Because what we have upcoming conversations on is a little simplified head of discussion. And what's that? Yes, that is the naming technique of the monoclonal antibodies. I will be doing this in short, yes, and I may repeat sometimes in future in details, but now without any delay, let's get into this. Now, did you notice that antibodies, all that you have heard till date, they end with MAB. Now, let's break the whole name of antibody. Letter just before the MAB, that is MAB, it indicates sources of the antibody. Okay? Now, I know you cannot understand by just simply defining all these things. You need examples. So, at this particular moment, everything gonna be crystal clear when I will give you the examples. See, when I say OMAB, O means I have added a O letter in front of the MAB. It's a prefix. So, that stands for murine, as in the case of OMAB. Now, in place of O, if I replace this O with XI, then it becomes chimeric, partly mouse, partly human. XI stands for chimeric, and then it is definitely defined or it is pronounced as ZMAB. XI, MAB. Got it? Now let's replace this XI with ZU. ZU. That is humanized. And humanized, what do I mean by saying humanized? It's not actually fully human, nor it is fully murine. It is more of a human and less of murine. It is humanized. Got it? As in the case of ZUMAB. ZUMAB. Now, if we replace this ZU with U, only U letter I'm talking about, then it becomes UMAB. That is fully human. Yes, that is fully human. Now, I have told you one letter just before the MAB. Now, if there are more of the letters before the last letter that I talked about, then that particular thing or that particular set of letters or letter, it indicates the therapeutic use of that antibody. Now, again, you just cannot just understand all these things without the examples. And we do need some good examples to make our understanding crystal clear. So let's get to know them. Say, when it is the letter TU, that stands for tumor. When it is the letter VI, that stands for virus. When it is CI, C, that is standing for some circulation associated antibody. Now, I will give you some specific antibodies at this particular moment so that you can just recollect, refresh, rejuvenate your knowledge and connect with all the theoretical and the defining things that I've just narrated to all of you. Right? Okay. And sometimes what happens, there is no prefix at all. Then that particular structure or the product is denoting an immunomodulator. Not more of an antibody. It is more of an immunomodulator. No prefix, nothing. Then it's an immunomodulator. I hope you got this whole concept. Now let's spill over the discussion latte over the examples without any delay. See, first one, muromonab CD3. It is a murine anti-CD3 monoclonal antibody. Why it is murine? Because in front of the mab there is O. O stands for murine. Got it? It is used to prevent the transplant rejections. Let's talk about the next example. Another one is the rituximab. Now we have XIMAB, so that is a chimeric monoclonal antibody. It is used for the treatment of Hodgkin's lymphoma. Okay, another one. Palavizumab. Yes, there is ZU, so it is a humanized monoclonal antibody. It binds to the fused part of the respiratory syncytial virus and it prevents the infection. There is a VI again. Palavizumab. So there's a VIZUMAB. So VI is associated with the virus. Okay? Next one, bevacizumab. There is a VI, there is a CI, and there is Zumab. So it is a humanized monoclonal antibody, and it is, because there's a CI, so it is anti-VEGF antibody used in the circulatory disorders of the eye, and it's a very important one. Another one we have ranibizumab. It is another anti-VEGF antibody, and then I'm going to talk about adalimumab. Because it is mumab, there is a U in front of the MAB, so it is a fully human Ig capital G1 monoclonal antibody. Specifically, it binds to the tumor necrosis factor, abbreviated as capital TNF alpha. And it is, if you just remember or just for your general knowledge, you should know it is given by subcutaneous injection. Adalimumab, I'm talking about, okay? Now, these are all my talks of the day about the monoclonal antibodies. Hope you found it as interesting as I loved narrating all this stuff to you. You know, good conversations, they are as stimulatory as a black coffee and just as hard to sleep after. Now, I'm not inspiring you to not to sleep after. You just have to be a good listener, and truly, I just want to say, to be interesting, you should be interested as well. As I am today, all in full spirits, but wait, finally, before we verbally depart to talk again in the next episode, just a little piece of advice. Good laugh and long sleep, they are the two best cures for anything. Hope you got the hint. Okay. For all the updates and latest episodes of my podcast, please visit www.ispharmacologydifficult.com, where you can also sign up for a monthly e-newsletter of mine. It actually contains a lot of updates about the medical sciences, drug information updates, and my podcast updates also. You can follow me on different social media handles like Twitter, Insta, Facebook, and LinkedIn. They all are with the same name. It's Pharmacology Difficult. If you are listening for the first time, do follow me here, whatever platform you are consuming this episode. Stay tuned. Do rate and review on iTunes, Apple Podcast. Stay safe, stay happy, stay enlightened. Thank you.